This is the last section of Unit 2, and it's 2.7, and it's microorganisms' genetic control of metabolism. Put simply, this section is really on genetic engineering, which you'll hopefully remember some of that from National 5, and we'll go back over that to begin with. So the previous section was environmental control of metabolism, and in that what happened was humans provided the conditions for microorganisms to grow, and we had our sort of phases of microorganism growth. But although humans provided the optimum conditions, it was a natural process, I suppose. What's happening here is when humans sort of interfere with the metabolism of microorganisms, and I guess the best example of a product produced through this is insulin. So the learning intentions for this, first of all, explain how wild strains of microorganisms can be improved. Recombinant DNA technology, explain what that is, and then be able to describe the stages of genetic engineering, how DNA is transferred to microorganisms. And there's a number of words in here which will be new, and you need to be able to know, you need to be able to explain what each of these means and use them when describing genetic engineering at a higher level. So first of all, how can you improve microorganisms? So these are wild strains, just means what you would find naturally everywhere around you. And your options are mutagenesis, which is a means essentially to induce mutations, to make mutations happen, and then recombinant DNA technology. And recombinant DNA technology, recombinant just means kind of new, I suppose. So it's genetic engineering, for want of a simpler term, genetic engineering. So mutagenesis, as I said, the process of inducing mutations, and you probably know this yourself. What can be done, or what can cause mutations, and certainly in humans, this is things that can cause cancer and so on. So your two options really are ultraviolet radiation, and then also some chemicals can induce mutations. And just to show you this, there's a nice YouTube video you can have a look at, and that's the effect of UV radiation on human skin. So not necessarily radiation to the level that would cause mutations which would lead to cancer, but just how sunlight can change people's complexion. So you can have a look at that. So that's the video that I mentioned if you want to have a look. Okay, so before we get into the sort of genetic engineering at a higher level, I want you to see what you can remember from last year. If you haven't done Nat 5, then obviously you're not going to be able to do this. But although looking at those drawings, you probably would be able to have a rough idea. So what I'd like you to do is put the letters in the correct order. So this is the stages of engineering, genetic engineering all mixed up. And once you've put them in the correct order, a wee explanation or a wee description of what's happening. So press pause and have a go at that, and then we'll have a look at what you should have. So this is the first part of it. So first of all, we should just, what are we looking at here? On the left hand side, this up here, this would represent a human cell, so very simple of course, and lots of organelles missing. But this would be a human cell, and that'll be a chromosome, and then the green bit would be a gene of interest, perhaps the gene which makes the insulin protein. On the right hand side here, you might have worked this, well it does say there I suppose, this is a bacterial cell. You've got no nucleus, and the DNA is either in this chromosome or in this plasmid. So the first thing you would do is you would take out the chromosome from the human cell, which has got the gene that you want within it. Meanwhile, you'd find a suitable bacterial cell, and you'll start to come across this word if you hadn't heard it before. So a vector is what the bacterial cell is in this instance. So it's So the second part, this was E, And what you've got is the human gene being extracted from the chromosomes rather crudely. We've just got a pair of scissors here, but the human gene is cut out, and this is done using enzymes. Meanwhile, you've taken out your plasmid from the bacterial cell. The next stage is A, and the plasmid is opened up and the human gene is put into it, sealed in using, again, enzymes. So what you've now got here is a bacterial cell, or a bacterial plasmid, sorry, which has a human gene within it. Next up is C. So C, the plasmid is now modified. We can now say that this is genetically engineered. It's put back into the bacteria. And finally, you give the bacteria the conditions it requires to grow, and you produce a large number of it, and that will now have the human gene. And if you've got the human gene, it should ultimately produce the human protein if the bacteria has everything required in order to achieve that. So that's your National 5 level. So now we're looking at higher level. So in higher level, again, just don't let this phrase throw you. Recombinant DNA technology means genetic engineering, and as a definition of it, it's a transfer of plant, potentially, or animal genes to microorganisms, which then produce the animal or plant protein. And as hopefully you'll know, the reason why you do this is because bacteria or microorganisms can reproduce very quickly, which means that you can produce large amounts of the wanted protein in a relatively short period of time. So the stages here, I'm using the same drawings because the process really is the same. We're just going to introduce a few new terms and go into a little bit more detail. So to begin with, again, it's this section of this part is essentially the same. So the chromosome is extracted from the donor organism. So we're not necessarily saying human or animal here, whatever it is you want the gene from. Chromosome extracted from donor organism and required gene located, and you have your suitable bacterial cells identified as well. Stage number 2. So here's where things change a little bit. So the required gene is removed, and we said at Nat 5 level this was using enzymes. We're now going to name the enzyme. So the enzyme is called a restriction endonuclease, and this leaves sticky ends on the gene sequence. So a sticky end here, sticky end here. I'll explain in a minute what that means, but that's what's happening in terms of the animal or plant gene that you want. Meanwhile, the vector, and in this instance, it's going to be a plasmid. So I've used the bacteria as an example, so it's a plasmid for bacteria, or it could be an artificial chromosome. Okay, so you then now got your vector, and you've got your gene cut out. And here is your plasmid, so vector. So the vector is opened up, and the point at which the vector is cut open, that's called a restriction site. So the vector is opened at the restriction site, and what you're using here is the same enzyme, so the same restriction endonuclease as you used to cut the gene out of the chromosome. And the reason why you use the same one is to ensure complementary sticky ends. And this is what we mean here by complementary sticky ends. So what you've got here, you've cut this, imagine this is the plasmid or the vector. So you've cut this out, and what's been left in the end is AATT. Then down here, we've got the end of the gene. So you can see the gene has been cut in a way that means that you're going to have these bases able to pair up. Clearly, if that had been cut, say, AATTC, but this had only been cut to leave AAT, then it's not going to stick together properly. So that's what it means by complementary sticky ends. You use the same restriction endonuclease enzyme in order for the gene to insert itself correctly into the plasmid. So you've got the restriction endonuclease has cut open the plasmid and it's cut out the gene,
And the next section, or the next part of it, is the gene is then sealed in, and the enzyme that does this is called ligase. And you've encountered ligase before, haven't you? You know, we often talk about it as a bit like glue, and we came across that previously with the DNA replication and the lagging strand. So same sort of idea here. The DNA has been joined together using the ligase. And then finally, or not quite finally, but we've got the modified vector inserted into the bacterial cell. Okay, and that leaves us now with our genetically engineered organism. Something that came up before, we did say that you could have artificial chromosomes, and so you will, you know, often the vector is plasmids. However, when you've got particularly large fragments of DNA, what it might mean is the plasmid can't cope with that. So that would be an instance in which you would use an artificial chromosome rather than a plasmid for larger genes. So have a break, go away, and then come back maybe tomorrow and see what you can remember. So to begin with, I've said use a whiteboard pen, but whatever you think. So try to write it on a bit of paper, I suppose. So use a whiteboard pen to show the stages. So they should, you know, I think I had 5, so you maybe go for 5 as well. The 5 stages of transferring a gene into a vector, and some hints down here. So things you should mention are restriction endonuclease, ligase, vector, restriction site, and complementary sticky end. So press pause and have a go at that. So the stages you should have, this is the first one. So chromosome is extracted from the donor organism, may well be human, and required gene located. Meanwhile, the bacterial cell carrying the plasmid is identified. Number 2, the required gene is removed using restriction endonuclease enzyme, and that leaves sticky ends on the gene sequence. And you've also got your vector, so that would be a plasmid if it's coming from bacteria or potentially an artificial chromosome. The third stage is to open up the vector, and that is the part of the plasmid at which that happens, or on the artificial chromosome, is called the restriction site. And you use the same enzyme, the same restriction endonuclease, so that you've got complementary sticky ends. Just remember, this is what we mean by complementary sticky ends. So the reason why you use the same enzyme is so that the gene will match the open base pairs will match up with the open base pairs on the plasmid, so they can come together and bond. At the complementary sticky ends, that's the ligase enzyme seals the gene into the vector. And finally, you've got your modified vector inserted into the bacterial cell. So that's your stages of in recombinant DNA technology at a higher level. So there's just a wee bit more detail we want to add in. So describe the features of a vector suitable for recombinant DNA. And number 5, explain why in some instances DNA is inserted into yeast rather than bacterial cells. So here is a plasmid, and in order for it to be suitable, you need to have various features. And the first one we've already mentioned, and that is a restriction site. So that is where the vector is cut open and where the gene is sealed in. And you also need these two features as well. So what's called an origin of replication and also a marker gene. And the marker gene, for example, could make it resistant to ampicillin, which is an antibiotic. Remember, antibiotics kill bacteria. So restriction site, we've mentioned that before. So that is where target sequences of DNA are cut by specific restriction endonucleases. So you need to have that. Secondly, we have an origin of replication, and that allows the plasmid of the artificial chromosome to self-replicate, to copy itself.
And finally, you want a selectable marker. So what it does is it protects the microorganism from selective agents, so that is antibiotics. And after exposure to the antibiotic, only microorganisms which have taken up the vector will grow. So just to explain this, let's imagine you've got a dish which has got, or an agar plate with your bacteria in it. Now, the bacteria that you've genetically engineered. Now, some of the bacteria might not take up the, the gene that you wish them to. And what the selectable marker allows you to do is to distinguish those which have taken up the gene and those which have not. So if they have taken up the gene, and if they're genetically modified, then they should be resistant to ampicillin because they've got the marker gene. So what you then do is you can then apply the antibiotic to all of those microorganisms, and only the ones that are genetically modified are going to survive because they're the only ones with a marker gene. So you expose the antibiotic to the microorganisms. Those that maybe it failed for whatever reason, they're going to die because they don't have that marker gene, whereas the ones that are genetically modified are going to survive. So you end up with only genetically engineered microorganisms. So that is the point of the selectable marker. Something else to mention is regulatory sequences. So they control gene expression. So remember, gene expression, if you've got genes switched on, then they will go ahead and produce the protein. So you need to have them in order for the bacteria to produce the protein that you want, whether it's insulin or whatever else. And something else that might happen as well if you've got genetically modified organisms. I mean, legislation is different in different parts of the world. Genetically modified food is pretty common in the UK, but sorry, pretty common in the USA, but not common, I think, in the EU or the UK unless it's for animal feed. So there are safety measures in place. So, for example, extra genes might be introduced so that the microorganism can't survive in an external environment. So, if it was to escape from the lab, or be taken away in someone's clothes to their home, a lab worker, then they shouldn't be able to survive. The second part we mentioned was why you might want to use yeast rather than bacteria. So if you remember from before, bacteria are what are known as prokaryotes, and we animals are eukaryotes, or we have eukaryotic cells. So if you've got a eukaryotic gene and you wish to produce active protein, then sometimes what you need to do is, well, it won't work in bacteria, they're too genetically different. So you would need to use a eukaryotic microorganism, and the eukaryotic microorganism that you would use would be yeast. So this produces active proteins. So as I was kind of alluded to, if you've got bacteria, then that is a prokaryote. So plant or animal genes, eukaryotic genes, expressed in prokaryotic microorganisms like bacteria, you get your polypeptide chain, but it doesn't fold correctly. So hopefully you remember this from Unit 1. What happens? You've got your chain of amino acids to make the polypeptide, to make the protein. However, the protein doesn't actually become active until it folds in a correct manner. This was something that was in the news quite recently, actually, that it was very difficult for scientists to work out how proteins folded correctly to become active. And I think it was artificial intelligence that worked out how these proteins folded. But yeah, that's why you would perhaps use yeast rather than bacteria, so that you end up with the correct folding. Okay, time for another break, and we'll just have one more section, one more lesson on this, one more part. Okay, to start off with, I'm hoping that you can remember the stages now and talk about it in sufficient detail. In the last lesson, we went through what you would want to have in a suitable vector for recombinant DNA technology.
And we went through all of these, so see if you can press pause and see if you can match up the correct letter with the correct number. So the first one, A, restriction site. That is where specific endonucleases or restriction endonucleases cut. And they are enzymes, remember, so that's within the vector. Origin of replication allows self-replication of vector, so B is 3. A selective marker 2 protects organism from a selective agent, e.g. antibiotics. D, regulatory sequences, they control gene expression. And E, finally, extra genes, they may be inserted in order to prevent survival in an external environment. So E is 1. So last little bit in this, just have a read through this. There are 10 mistakes and I want you to, just one word mistakes, highlight them and then write down what the correct word should be. Okay, press pause and do that. So first paragraph, genes inserted into recombinant plasmids are artificial chromosomes for smaller genes. That's a mistake. So if it's an artificial chromosome, then you've got a larger gene. So these genes are used to improve wild strains of microorganisms. For safety, extra genes may be introduced to prevent death of the microorganism in an external environment. So it's not to prevent death, it's to cause death. So to prevent survival would be the more appropriate word in there. To be suitable for gene insertion, recombinant plasmids must contain a restriction site, introductory sequences, an origin of duplication, and a marker gene. So that should be regulatory sequences, and then that should be origin of replication. Okay, so regulatory and replication should be in there. The restriction site is where the plasmid is cut open by ligase. It's not by ligase, that's the next one there is by restriction endonuclease. The origin of the duplication controls self-expression, so that should be replication. And I don't think self-expression is really a priority for bacteria. That should be gene expression. So the origin of replication controls gene expression. A marker gene is used to ensure that the fragment has been successfully inserted into the vector, e.g. a gene for herbicide resistance. So herbicide resistance would mean that you're not killed by weed killer, which is not really for bacteria. What they would want to be resistant to is antibiotics. That should be antibiotic resistance. So we've had 1, 2, 3, 4, 5, 6, 7, 8. So 2 to go. Eukaryotic genes must often be inserted into a eukaryotic microorganism. Archaea, it's not archaea, it's yeast. This is because eukaryotic genes expressed by prokaryotic organisms, e.g. bacteria, can result in proteins with incorrect transcription. So it's not transcription, I think the transcription producing the messenger RNA is not a problem. The problem comes when the protein has to fold and become active. So that should be incorrect folding for the last one. Okay, press pause and have a go at this. So for recombinant DNA technology, I'm not sure if there's, I suppose there is a potential maybe for it to be a kind of 8 or 9 mark essay question if they were to ask you to go into detail about features of a suitable vector, but certainly if you were to go through the stages, then it might be a 4 mark question. So press pause and try that. So for your 4 marks, there's quite a few things that you can say, and just the first one is just to say what it is. So it's when a plant or animal gene is transferred into a microorganism so that the microorganism makes the plant or animal protein. Okay, I know a lot of you will probably just jumped in there to get onto the stages, but it's always worth just saying that what is recombinant DNA technology. So second mark, the restriction endonuclease is used to cut the gene out or to cut open the plasmid or the vector. And then the next part, the genes are introduced to increase yield or prevent the microbe surviving in an external environment. 4, ligase is used to seal the gene into the plasmid. 5, recombinant yeast cells to overcome polypeptides being incorrectly folded. Forget about that second bit lacking post-translational modifications. That's been removed from the course. This is a kind of simpler way if you had just said that yeast may be used
so that the proteins are folded correctly. That would be okay for this one, yeast rather than bacteria is used. And then finally, regulatory sequences in the plasmids or artificial chromosomes are used to control gene expression. So you can see if you kind of went through the stages, you'd maybe get potentially 2 out of that. What they're looking for is a kind of detail about your regulatory sequences and why you might use yeast, your selectable markers, that kind of aspect of it, and naming, restriction endonuclease. So just be careful for this. You don't give an answer which is about 2 national 5. And this is if you have to have a look at this, genetic engineering will change everything forever. This is a nice little video just to give you an idea of where this all may be heading if you want to have a look at it. So that is us finished Unit 2. So as I like to do at the end of units, you could have a go at this. I've just picked out some of the words, sort of things, words you need to be familiar with and relate to biology. So you can have a go at this as a bit of revision of Unit 2. So press pause and then I'll give you the answers after that. So anabolic, this is from right at the start of the unit, so it's a reaction in which energy is used to build up larger molecules from smaller ones. Competitive inhibition, so that is when an inhibitor binds to an enzyme's active site, so make sure that's the kind of key bit, prevents the substrate from binding. You might have also said that competitive inhibitors reduce the rate of reaction. Glycolysis, so now the next 3 are going to be from respiration. So that is the first stage of respiration, and it's when glucose is broken down to pyruvate in the cytoplasm. Dehydrogenase is an enzyme, and it removes electrons and hydrogen ions from various compounds during respiration, and when it does this, it turns NAD into NADH. And remember, NADH is used, goes on to the electron transport chain, and carries hydrogen ions to the electron transport chain, which leads us into the next one. So it is the final stage of respiration. We're on the inner membrane of the mitochondria, and kind of key, there's quite a few things you could say here, but the kind of important bit is you're producing a large amount of ATP, specifically 34 ATP molecules, and it's ATP synthase that is involved in doing that. A regulator, so that is an organism which uses metabolism to achieve homeostasis, so that maintains a stable internal environment. So, the example is if you become too hot or too cold, your hypothalamus detects that, and then you have these kind of negative feedback mechanisms which result in sweating, or you're losing heat because blood goes to your skin and so on. And then hypothalamus, that is, as I said, monitors your body temperature. Aestivation, so that is a type of consequential dormancy which animals carry out in order to survive excessively hot and dry conditions. Then moving on, secondary metabolite. So this is the last 2 are from the last 2 parts of the unit. So this is produced during the stationary phase of microorganism growth, and it's not essential for growth, but it might give the organism some sort of advantage. And finally, restriction endonuclease. That is an enzyme, and it cuts open DNA sequences on genes or at the restriction site of vectors, and it produces complementary sticky ends so that the gene can be successfully inserted into the vector.